All right, so following a quick technical difficulty, we're finally in the podcast. But welcome back everyone to the Synthesis Workshop podcast. Today I'm delighted to be joined by Professor Abby Knight, who is at UNC, and a lot of polymer chemistry, a lot of DNA polymers, and I'm really excited to have her on to discuss that today. But before we get there, we'll definitely start with, your background, have the audience try to get to know you here a little bit. But I was like reading through, you had done your undergrad at UNC, and you returned now as professor, but perhaps we could start there. Did you grow up in North Carolina, or kind of from that general area?
So I guess I'm from the general area. I grew up in Charlottesville, Virginia. And because Charlottesville, Virginia was my hometown, I wanted to broaden my horizons as an undergraduate.
Sure.
And one of the positives of UNC is that it is significantly state supported. The tuition is exceptionally reasonable for both in-state and out-of-state students. So yes, I was an undergraduate here, and now following my PhD and postdoc, I'm back.
Awesome. You said Charlottesville, Virginia, right? I need to pull this up. I need to get a map because I'm from Pennsylvania, but I always kind of need to center myself. Let's see, where are we looking at here? Isn't there a huge school that's there in Virginia? Is that
the University of Virginia is there. Yeah.
You didn't want to go to the great University of UVA?
I wanted, I believe I told my parents I did not want to run into my high school teachers in the grocery store. Just wanted something a little bit different.
What is it about that is so awkward? I don't know why. Even when I was in high school, why is it so weird to run into professors or teachers outside of, I don't know, maybe it's a weird concept that teachers have lives, I guess. I don't know what it is.
I don't know. Yeah, but I've heard from graduate students here that they have ducked behind shelves in order to avoid seeing faculty in grocery stores. So I think it continues.
I mean, to be fair, I guess there is something about leaving work at work and when you're home at home. So I do get that, but I think people take it a little extreme sometimes. So, it's like we're just people too at the grocery store, whatever. But I've actually never really traveled to Virginia, so I haven't I know DC's right in there. I think that's technically Maryland though. But in any case, I don't know if you want to tell us a little bit about Charlottesville. I mean, what's there to do there?
So here's Virginia. The DC area is all of Northern Virginia and Maryland surrounding DC. I would say is very dominated by that culture. Charlottesville is in the middle of the state. It's about an hour from Richmond. It's very much a college town. Honestly, Charlottesville and Chapel Hill are very similar. Something I like about Chapel Hill is that we have the proximity to Durham and Raleigh. They're a little bit closer than more urban areas are to Charlottesville. But Charlottesville has quite quick access to the Blue Ridge Mountains. You're easy driving distance to Virginia Beach and even the North Carolina beaches.
Virginia Beach, let's go.
Yeah. But yeah, honestly, pretty similar, very similar weather, pretty similar culture.
Right. So, kind of coming up through, elementary all the way through high school before attending UNC, did you kind of have any general interest in STEM, or, what were kind of your interests and hobbies kind of coming up?
Let's see. So I have always been interested in STEM. Even, I was super little, I liked math. I think the first thing that I wanted to do when I was little was I wanted to work. I was really excited about space. This was back when Pluto was a planet. So, a little bit ago.
Oh, man.
I know. Being an astronaut seemed a little intense. And so I thought working at mission control was pretty much the ideal job. But as I got more into STEM, chemistry and biology aligned a little better with my interests in physics.
Fair.
So that's sort of what landed me here. I always, I feel like anyone interested in STEM is but maybe med school. And then every time I'd be that's really deeply not for you. I don't do well with blood.
Same.
Reasons.
I get squirmish looking at my own blood. Literally.
Yeah, it's not great. How could people do that all day? Whatever. We need doctors.
Yeah, right. We do need doctors.
All of you that do that.
Yeah, thank God. And yeah, it is kind of funny though. A lot of people who are on the like, let's say medical school track or dental school, PT school, whatever, they get to like organic chemistry and it's like the end all be all. We need you though. But sometimes in chemistry, we have to convert you guys, convert some of the medical doctors over to
people
the
just
STEM.
aren't as familiar, right, with the job options in chemistry. Like when you play the game
Right.
of life as a child, like doctor is one of the career options.
Right.
So I think, yeah, we get some people switching over mostly because of a lack of familiarity.
Right. Yeah, that's actually a good question though, because I hadn't really thought about it. Is like coming up. Has anyone told you like what a chemist does, or even like I guess, you know, professors, obviously because you're teaching, but like you're ultimately a teacher. So, but I mean, yeah, has anyone like told you like what chemist does? I don't know, because I haven't. I didn't until I got to college.
Yeah, even in college, I feel like it's hard to know. Like, I think we spend a lot of time trying to tell our majors about what the job options are. But
Right.
even, you know, when we get entry graduate students, right, you know, I'll ask them what they want to do, and they don't know, which is super reasonable. You learn a lot over the course of a PhD.
Mm-hmm.
And some will be like, I'll be like, okay, well, do you know what you don't want to do? No, I don't want your job.
That's it.
Oh. At least we've established
something. Right.
But you know, like I have friends from my PhD that went into policy, that are consultants, that are patent agents, and all of these, you know, need people with a background in STEM.
Right.
But yeah, it's super hard to know, I think, what those options are.
I guess the biggest thing is too is like when you think about I'm trying to think like when you in elementary school you have like people come in and tell you about what they do. I don't think I've ever seen a chemist before, but I don't even know how to. This is something we got to do better as maybe as a chemistry community, is like articulating what we do, and of course it's easy to kind of do demonstrations and stuff like that. But I don't know. I still feel like we got to get more out there, you know.
Yeah. So, communication is super important.
But yeah, so you know, you went to UNC, and you also did undergraduate research with Professor Marcy Waters, I believe. Do you want to tell us a little bit about how that opportunity arose and how you know, what you did in undergraduate research?
Yeah. So I was pretty undecided about what exactly I wanted to do in terms of research. And so when I started, you know, here at UNC as an undergraduate, I was a biology major. And I actually did research in a more biological lab the year after my first year, looking at some properties of Ebola surface proteins. And that, you know, I thought was very cool, but wasn't quite what I was interested in. And then I did some research with my colleague here, Ed Smolsky, who works, I would say, more physical chemistry, was working with liquid crystals. And that also wasn't exactly what I wanted to do. And I talked to, you know, my advisor, and he was like, why don't you try organic chemistry? And I was like, I don't know. Because it's really hard to tell from the organic chemistry class what organic chemistry is.
Right.
So like even after I teach organic chemistry now. But after taking the class, I wasn't necessarily sold on it. And I probably, honestly, after a few emails, was able to get in touch with Marcy Waters, and ended up working with one of her graduate students at the time, trying to develop these small molecule intercalators into DNA. And we wanted to append the DNA with something that would make it more specific for specific sequences. And we're using some dynamic library generating strategies to do that. You know, the techniques were really cool. I didn't end up publishing anything during my undergraduate research. But it's honestly like some of those ideas were the inspiration for some of the things that we do today.
Right. Yeah,
so still valuable.
Right, of course. I mean, any undergraduate research is like so invaluable. I'd even argue it's like, especially if you're going to go to graduate school, it's probably the most important thing you could possibly do because it kind of just gets your feet wet. And, you know, I can't praise my undergraduate advisor enough to kind of put me on this track in a sense. So, yeah, it's extremely important. It may not be groundbreaking. You may not well, you might solve cancer, who knows? But like, it's just about getting it's just about getting in there and familiarizing yourself with a lot of the techniques and just having fun. Honestly, it's just really just curiosity, I think. But so you came in as a biology major. Was it? I mean, did you have any idea what you wanted to do? I mean, obviously, it's hard to parse out. I mean, listen, at 18, 20 years old, I get it. No one really knows what they want to do. I get it, but for the undergraduate research, though, I mean, did you like ask the professors? Were they recruiting students? Like, because it takes some initiative, I'd say. Like, you kind of have to
get into a lab.
Yeah, I mean, you do.
I definitely. It is a dusty memory, I would say, those days of trying to get into undergraduate labs. But I'm pretty confident that the process was as sort of chaotic as it is now when I take undergraduate in my lab. I sent a bunch of emails, probably followed up on those emails, and then was able to find positions in these labs. And so I feel, very fortunate that those positions panned out.
Right. And what kind of, well, I guess you still do a lot of, biology now, but what was the initial interest? You were just kind of, I'll just do biology or as a major or kind of how
it's importance.
Right.
Like, people want to be doctors because they want to help people, right? When you see a biological question, you're like, oh, like curing diseases. That's like super easy to explain to family, to everyone, right? Like that's definitely important. Whereas now, my research is making these protein mimetic macromolecules. And people are like, oh, cool, but like, why? I go, well, we could solve all sorts of problems. It's a little harder to see the importance of chemistry research sometimes as opposed to biology.
I listen, I get it. It's definitely, chemistry in general is not quite as marketable, but we are working on it. Now, following your undergrad, though, you went on to do your PhD in chemical biology at UC Berkeley with Professor Matthew Francis. I don't know if you want to tell us a little bit about how you came to that decision, because graduate school is it's a very, very obviously very important decision. But was it a natural fit to do next, or kind of how did you come to that decision?
So I have always been interested in being a professor, and I don't think honestly, I don't think I knew really what that job meant until like now, like years into being a professor.
Right.
But graduate school definitely seemed like a natural step. I really liked doing research. And my undergraduate advisor was super helpful in talking through both different schools, but also faculty that she was familiar with that she thought could be a good fit for my research interests. I'm pretty sure Matt Francis was on her list. But I also, I didn't go very far away for college. I thought it would be really fun to go to California for graduate school. UC Berkeley is a great option. And so I think the combination of, there was a lot of exciting research going on there, and the location being, so different and exciting, that is where I landed. And then when I was there, I was in a chemical biology program where we did rotations. And so I got to experience a little bit, being in the Francis lab, before I joined.
Yeah. How do you kind of like the rotations idea? Cause I didn't get that here in Houston, but I don't know, like, I know a lot of schools, well, really in general in California, I feel like they do a lot of rotations there. That's kind of like almost normal. I don't know how you
common for like biochemistry type programs. Okay. But I am a big rotation enthusiast.
Fair.
So we didn't do rotations across our department when I started. And so I have implemented a rotation program in our department, which started with just a few sort of subsets, divisions in the department. Now we do it department-wide. It's obviously not perfect. There's definitely some downsides, but I think the things that are really positive about it is you do get to, trial being in a lab, see what the culture is like. We do so many different techniques in my lab that I tell students who are rotating, like, if you do solid phase synthesis and you realize that makes you want to jump out a window, like, that's not the project for you. And now we know that. So I think that's valuable. But broadly, my students get to rotate, the ones that join my lab get to rotate in other labs too. And that, I think, it builds these bridges in the departments such that, they're naturally talking to other students about their research that's ongoing in my lab. If we need some instrumentation or they need advice on, will my lab run gels? And I hadn't run a gel in like a decade. I think that network is beneficial for the department as a whole, in addition to, giving students and faculty the opportunity to learn more about each other in the process.
Right. Yeah, I definitely think the positives there, in fact, I don't even know if there are any. I've never participated in rotation, so I don't even know what the negatives are, but it seems like the positives are just, they'd way outweigh them anyways.
I agree with that.
But yeah, so I guess if you want to tell us a little bit about the research that you did as a graduate student, because I was looking through. I know you were like looking at ligands to do, look at was like water and environmental remediation. I went deep into the publications, and I saw one of them was like extracting like chromium-6 or chromium-4, I think, right, from water. I think, I don't know. If you, I'd love to hear about what you did.
Yeah. So the Francis Lab, their main area of expertise is in making protein-based materials, and specifically the development of bioconjugation reactions.
Okay.
And so that is a lot of what my peers did. But we had sort of this exciting opportunity that was industrially funded and collaborative to develop materials that were useful for these industrial type separations. And so, when I met with Matt and was joining the lab, he, introduced this project to me where we would try to make these peptide-mimetics for these types of applications. And the reason that we ended up focusing initially on hexavalent chromium as a model system is that reference gets more and more distant with time, but there was this movie, Aaron Brockovich, which talked about this incident decades ago at this point, but PG&E in California had a lot of hexavalent chromium go into the groundwater. And if you dig deep into that sort of environmental crisis, you'll realize that despite it being heavily popularized with a movie about it, there was no real solution. The solution they ended up going with was letting it dilute over time. So there are no cost-effective solutions for this type of remediation. And I think that I haven't done the deep dive into Flint, Michigan, but it's a similar type of conundrum where it's incredibly important, affecting a lot of people and communities. But we just don't, it's a somewhat challenging problem to solve. And so we made these peptide-mimetic libraries in order to identify materials. I always get the thumbs up emoji because I move my hand
do you have a Mac?
much.
Do you have a Mac?
Yeah.
Oh my God. Okay. Yeah.
Is it a Mac thing?
It's a Mac thing, apparently. I was talking to someone on the podcast a few weeks ago, and this happened like 3 times during the podcast. It's hilarious, honestly. It's so hilarious.
Oh my gosh. Okay. Anyway, so libraries bind hexavalent chromium. If you can bind it selectively, then you could use a limited amount of material to do these separations.
Okay.
And so that was our, mission. We identified some of these peptide-mimetics, and it was quite a cool project. It was again sort of in this high throughput realm, which is something that is important again to my lab now. I ended up talking to some companies. After our paper came out, there are all sorts of different interests in the technology, but peptide-mimetics can be more scalable than peptides, but are perhaps not easily adaptable to our existing infrastructure.
Sure.
And so that was part of my motivation to do a polymer science postdoc, is that, those are the types of materials that can be easily adapted or more easily adapted to this scale of challenge.
Right. Just to kind of maybe give the viewer a little bit of context for it, because I'm unfamiliar with it. I mean, and I know this is not, perhaps your background necessarily, but do you know where like this hexavalent chromium was kind of coming from? Like what Okay.
Definitely used to. I
That's okay.
can't remember what PG&E was using it for.
Mm-hmm. That's
Um,
okay.
um,
I mean, people can definitely just look it up too. I'm just curious.
I
Um,
should know.
it's definitely, well, I mean, it's a long time ago. But um, yeah, it's definitely very toxic. But the principle of what you're doing is super cool. Was there a general, I don't know, perhaps you could maybe tell us a little about kind of the outcomes. Like, I mean, what did these, ligands look like? And
What were we able to do?
yeah, like
Yeah.
what were some of the outcomes for it? Like kind of big picture stuff.
Yeah. So we made peptide-mimetics. That means they have the same kind of fundamental backbone as peptides,
Mm.
components of proteins, and the ones we used were peptoids, where the side chain, instead of being on the carbon in that peptide backbone, is on the nitrogen. We made relatively small materials, so oligomers, mostly sort of 4 to 6 monomers.
Okay.
And the end of the paper you're referencing, we did this kind of cool experiment, which I believe was my advisor's idea, where we took some natural water, both from this creek that runs through the UC Berkeley campus, like pictures of my undergrad and I collecting those water
Oh, that's
samples,
so cool.
and then from the beach, so some ocean water, to
Hey.
see with these real water samples, can we pull, like we doped in the hexavalent chromium. These are not contaminated sources.
Right.
We doped in some relevant concentrations. We were able with our materials to pull out enough chromium that it was down to below the EPA regulations
Oh, wow.
for not everything but seawater. Seawater, the level of those competing ions was significant enough that we weren't able to significantly decrease it in some conditions. But
Right.
in most drinking water-based sources, we were able to pull it
And
out, which was really cool.
that's still super cool though. Yeah, I mean, but it's probably where you'd find, well, ocean water is obviously really important, but creek and freshwater sources are arguably,
Yeah.
where, and that's really where we get like our drinking water from. So those are definitely,
Yeah.
that's super cool. Has it, since you've kind of left the project and kind of started your independent career, do you know like where it's kind of gone since then, or what was kind of the,
That's
I
a
don't
good
know.
question.
Maybe I'll have to follow up with him on this.
Yeah, you should invite him on.
Yeah, this just might be on.
I do think there's been some other funding. I don't know. To my knowledge, I don't think I've seen publications after mine. It was like sort of a fringe area of the
Sure.
lab.
Yeah.
And
That's super
something that, yeah, we're not using that exact system, but we're still interested in these industrial separation types of problems in my lab now.
Yeah. Okay. All right. So we'll definitely put a pin in that, and we'll kind of circle back to this in a second. But okay. So one last thing before we move on to your postdoc. General, when you're not inside the lab, general activities. I always love asking people what they did at Berkeley, because I always seem to get a different answer. Obviously the nature is great there. I don't know, like the ocean's fantastic.
It's such an amazing place to live.
But what were some of your favorite things to kind of do as a graduate student there?
So the Francis Lab had an annual ski trip. So I had never skied before, and I cannot say I'm good at skiing now. But I did some skiing while I was there. I think, growing up in a college town, being in a much more urban area, like there are just the diversity of food options, right? Just going out to dinner with friends, and getting to know my like relatively large cohorts, was really fantastic.
Awesome. So, from graduate school, you moved on to become a postdoc, staying in California, right? So now you're at Santa Barbara with Professor Craig Halker. You had mentioned that had already thought about becoming a professor. So perhaps I assume this is kind of just a natural progression then. I don't know. But what kind of drew you to go into Professor Craig Halker's and head to UC Santa Barbara?
Yeah. So I was telling the story earlier today. that I am pretty sure that when I talked to Matt about postdoc options, he was like, "Oh, I think you should really consider doing a postdoc with Craig Hawker." And I was like, let me make my own decisions here. I'm going to do my own research. I'm going to come back to you with the results of that sort of deep, thoughtful process. I like walked back into his office and I was like, I'm strongly considering working for Craig Hawker.
Sometimes your advisor knows you better, I guess, than yourself sometimes.
Yeah. 100%. My appreciation for both Matt and Craig has only grown with time. So yeah, I think, it's a natural, a lot of students from the Francis lab had some more material type interest. I think it's a natural progression, but it was a great fit for me in terms of both the science and the community of the lab.
Mm-hmm. Actually, one thing, what was some, like when you're considering postdocs, because, when you're a grad student, you obviously you basically get 5 years of skills and you become an expert kind of in your area. And a postdoc really is an opportunity for you to kind of, become your own thinker and learn new skills. So what, going to your postdoc, what were kind of the techniques that you wanted to step out of, or like, that to like learn about really?
So some things that I was really excited about Craig's lab was definitely like, it was not only that polymer science is more applicable, easily translatable to industrial problems, but, he had a significant amount of industry funding and those collaborations were pretty strong. So while I was a postdoc, I was able to go to the Dow site in Collegeville, right, and like directly interface with industry scientists.
Wait, Dow, like Collegeville, Pennsylvania?
Yeah.
Oh, okay. I'm literally right from there. That's where I grew up. I grew up
really?
right in that area. Yeah.
It was surprisingly lovely.
Collegeville,
Pennsylvania.
Yeah. It's so rare. Like a quick aside, Pennsylvania, Southeast PA has so much like industry there. Yeah, I mean, Dow, it's so much there. Like for like oil, gas, and energy. Just a side note for anyone, like take a look at Southeast PA. There's a crazy number of industry there. But anyway, I digress.
Nice.
And it is nice. Southern PA, yeah.
But yeah, so I think, that was a strong draw. Being in a large lab as a postdoc, there were more than 10 postdocs when I started in Craig's lab. And so having that community was really fantastic, right? Like these were scientists from around the world that, and all sorts of different areas of expertise that were at the same career stage as me, and they're still some of my closest friends. So I think that was an additional benefit to the lab.
Right. And what were you, what were you working on as a postdoc?
Yeah. So as much as I say, I was involved in some of these collaborations with Dow. The one with Dow Collegeville was specifically looking at paint dispersants.
Mm-hmm.
But the project that was more, I guess, my initiative and something that I've taken into my independent career is not industrially applicable, but was sort of building off of these small oligomeric biorelevant chelators. I wanted to ask the question, if we take these small chelators and put a hydrophobic polymeric tail, there's a lot of work putting lipid-like tails. What types of things could we achieve? And when I wrote my postdoc fellowships, I wanted to be able to mimic natural enzyme sites or, chelators that had other applications. And that is something that we are still interested in doing, but have yet to achieve. It's been sort of incremental steps. So my work in the Hawker group, you're really ambitious as a postdoc. You're like, let me just go ahead, put together a few quick papers
right.
On something that I've never done before. It's going to be great. But basically, the quick summary of the project is we took a hexahistidine, three peptide or a peptide with 6 imidazoles in a row, put on this oligostyrene, the same monomer that's in a styrofoam cup, We self-assembled that. That self-assembles in water, driven by that hydrophobic tail. But what was kind of cool is we could add different metal ions that had different coordination geometries, and each of those would generate a different morphology.
Okay.
So with the same material, we could generate different assembled morphologies. So like, vesicle that has water inside, or a micelle with a more dense core. We had micelles that ended up connected together. There are all sorts of sort of cool things we could make with a single material.
Okay, so let me try and see if I get this, because this is not really my not really my area. But
yeah.
So you had oligomers made of peptides. No, wait, amino acids. I'm sorry, amino acids.
Yeah, so we have one oligomer that is amino acids,
right.
6 histidines in a row.
Gotcha. Okay, and without really changing anything, or when you change the metal that was introduced, that would take different shapes, like how it bound it.
Yes, but the histidine is bound to this hydrophobic oligomer of styrene.
Okay.
And so the hydrophobic oligomer of styrene is what's helping it self-assemble, just like soap self-assembles into a micelle. This hydrophobic component, those all condense together, and that gives you one morphology, but the metal ions, then when you added them, would give you different three-dimensional shapes.
Gotcha, okay. Now, is there kind of a general trend for the metals and shapes that you would get, or like what?
They correlated, so we couldn't concretely prove the coordination geometries, but the morphologies that we got did trend with the preferred geometries of the metal ions.
Okay. And, how strong were these structures, though? I mean, obviously, like, I don't know, like,
that's a good question.
Are they easy to take apart?
Not
I have no
idea.
Strong. Okay.
Yeah. So they're probably not dynamic.
Okay.
So once some materials, like a cell membrane in solution, those lipids are able to diffuse around. Sometimes you can have lipids that are coming sort of in and out of your assembly. They can go into solution. With our materials, it is unlikely that we were having any go into solution. You would need heat in order to allow for overcoming that energy barrier.
Mm-hmm.
But if you like if you sonicated it or something, right? If you tried to put it through some sort of filter, probably our materials would break down. Like they are driven by non-covalent interactions, and they're not particularly strong.
That's okay. Yeah, okay. I follow that. That's so really, really cool. Like, proof of concept, I guess. And then it's kind of seeing, we're still doing this now. So I think this is probably, good transition then into, your area now. So you're now a professor at UNC, and you really have a lot of projects going on here. So maybe perhaps I don't know. I'll let you kind of take the floor here and kind of let's kind of go through each area of your research, and we'll talk about an introduction maybe to each of them, and what are the implications of each, right? So what's kind of been learned so far, and where it's going. So perhaps you could start maybe with like the biomimetic copolymers. I don't know what, or actually no, let's take a step back. I'll let you explain your research.
I appreciate that. No, I appreciate you looking into it ahead of time. So broadly, we make protein mimetic copolymers. Our hypothesis is that proteins are made up of 20 amino acids. They're able to have exquisite control over their structure and function. With synthetic copolymers, we have comparatively infinite options in terms of the monomer scope. You can have all sorts of different backbones. You can buy all sorts of different monomers. And so in theory, we should be able to outperform natural proteins. But in the vast majority of cases, we cannot. And so, part of the reason for that is that the tools for synthetic copolymers are much more nascent, and both experimentally and computationally. But our hypothesis is that by learning more and taking inspiration from both chemical biology and these natural systems, we can develop more functional copolymers.
Okay.
And so the ways that we approach this are first sort of building off of my postdoctoral research. We still make these peptide polymer amphiphiles. They're oligomers. They're not very long, but we call them peptide polymer amphiphiles. And what we have done so far in that area is we have taken some, again, pretty vanilla peptides. They're not functional. They're kind of boring. We wanted some boring peptides to start to understand the scope of things that we could make.
Right.
And so we have shown with our first paper that we can make all sorts of different morphologies with these peptide polymer conjugates. No more metal ions, just looking at changing aspects of the peptide and the polymer. And then in our most recent work that we were submitted on, you asked sort of about a little bit about the dynamics of the assemblies that I was making in my postdoc. And that's something that's the focus of that work, where if you tune what that polymer is that's attached to your peptide, that can impact the dynamics of your assembly, which, I think makes sense if you think about it, but wasn't necessarily why we originally went down that path. And so that means when you take an amphiphile and you assemble it with different mechanisms, this sort of complex assembly landscape where, here you have one distribution and here you have one distribution, and how you assemble it helps dictate where you land.
Right. Okay. So I'm going to try and work through this a little bit. So, okay. So the peptide polymer amphiphiles can take on different morphologies. Now, okay, maybe when you're starting out kind of in this study, what sort of polymers are you working with? What's the monomer, and what sort of peptides have you looked at in the beginning here?
Yeah. So my postdoc work for the polymer, I use styrene, what's in styrofoam cups.
Okay.
Since transitioning here, we in our first paper used oligoethyl acrylate. It's just an ethyl chain as that pendant chain. And then we've sort of stuck with acrylates for the time being in our work in this area. So we just tried other alkyl acrylates in our most recent work. So,
Gotcha.
Cyclohexyl being kind of the most fancy.
Sure. But I guess, and you also mentioned too that they're because part of my research, not my research, but our research group is full polymers, and so when I think polymers, I'm thinking the metric ton scale. But we're talking about oligomers, right? Or okay. Now, do you have a general sense of how long the polymer or / oligomer chains are, or what?
Usually, so they're a distribution,
okay.
Right? Because we're making them with controlled radical polymerizations, and so if we were using a lipid, you get one molecular weight. If they're controlled radical distributions, we have more than one, and they're usually centered because we're using pretty small peptides. The degree of polymerization, so the average number of monomers is around 5 to 10, 15. So really small,
right?
Because our peptides are also pretty small. And right now the peptides we're using are designed to be unstructured, so that we don't have to think about how secondary structure is affecting the assembly while we're doing some of the foundational work.
Gotcha.
But that's the kind of thing we're moving into, is these peptides that have both structure and function,
right?
To ask questions both about the peptide. It's a cool question you could ask with these assemblies is, the self-assembly can tune, the properties of that polymer can tune the size of your assembly.
Right.
And the curvature there, how much space your peptides have that are attached, can affect their secondary structure. And so our hypothesis is that we can fine-tune the secondary structure of our peptides in the assemblies and start to ask questions about how secondary structure affects function, which is really hard to do with a protein. You can't just look at a protein and be like, what if it was less helical?
I can imagine that particularly when you start introducing the secondary structure, how this can get really complicated really quickly. But in so far though, the what kind of controls or dictates whether you get a micelle or vesicle or worm, what sort of polymer / oligomer do you need, and what sort of peptides do you need to kind of get these assemblies? What's kind of been what has been learned so far in this kind of area?
Yeah, so there's some fundamental polymer physics concept called a packing parameter that we can loosely apply to our system.
Okay.
So usually with your hydrophobic component and your hydrophilic components, we strive for around the same molecular weight. But the more hydrophobic something is, basically the more hydrophobic stuff you're going to have. And so if you think about the space that your amphiphile takes up, if you have a short little tail, then your peptide is taking up more space. And it looks kind of like a cone. And that assembles into a sphere. You can imagine sort of how a cone would assemble into a sphere. And that gives you a micelle. But as you start to increase it and it looks more like a cylinder, that cylinder self-assembles into vesicles, so something more like the membrane of a cell.
Okay.
And so those properties, there's more complexities to it than that, but that sort of loosely guides our
design. Okay. All right. Cool. Now, what are some kind of other areas in research that we're targeting here?
So the one you mentioned at the beginning, which we call, bio-inspired copolymers, that I think is where we have the most work so far. And there, our hypothesis is proteins have structure. That's important for function. Let's put some structure into synthetic copolymers and see the effect on function.
Okay.
And so we've done some work with we have a monomer that generates these beta sheet-like interactions, and we've characterized that, and then shown that we can pattern this monomer into blocks or domains that looks, doesn't look like a protein. We don't have perfect sequence control with polymer synthesis like you do with a protein.
Okay.
But that amount of control can still, getting back to my industrial separations roots, affect function. And so the model function that we're interested in is now not hexavalent chromium binding, but lanthanide binding, because that's another really important industrial separation.
Sure.
They're, in my iPhone, lanthanides are in all sorts of green technologies. And so what we showed is that by patterning this beta sheet structure generating monomer, in addition to just an acrylic acid, which is a dependent carboxylic acid, we could control the affinity and selectivity of copolymers for lanthanide ions.
Okay. I need this. Wait, can you explain that again? I'm having trouble following this one.
Okay.
Yeah, I'm not a big polymer science is still kind of. The polymer science to me is like a bit of a head scratcher. Yeah.
It's different. It's just different challenges.
Right.
And so, proteins. Monomer 1, monomer 2, monomer 3, et cetera, perfect sequence control.
Yep.
With copolymerizations, you are not adding monomers one at a time. Usually you're dumping them all in. They're polymerizing based on their rate of addition, and that's kind of controlling your sequence, but it's stochastic. You have all sorts of sequences in addition to all sorts of molecular weights.
Gotcha. Okay.
So we have control, but it's not comparable to something like a macromolecule.
Okay.
But what you can do is you can say, all right, what if instead of adding all of my monomers at once, I just add some of them? So I'm going to make a loop-like block that only has hydrophilic monomers, and I'm going to add that first. And then when that's done, I'm going to add monomers that generate this more structured block or domain. And so we add some of our monomer that makes those beta sheet-like interactions. We can add in some of our monomer that coordinates to our lanthanide ion. And then say, okay, this block that we just made is able to have those secondary structure-like interactions in addition to coordinating our metal ion.
Okay.
And so in that way, we can control whether the coordinating group is in a structured domain or a loop-like domain, and we can control some other aspects of the sequence. And we show that those aspects that we're able to control can affect the affinities. These are all compositionally identical copolymer ensembles, right? They're still not a precise sequence,
Right.
but we get different functional outcomes.
I'm sorry. That makes sense to me. So do you guys then have like a target kind of structure in mind, and then you go into the lab and kind of build it? Or like what kind of comes first then? Like do you see, oh, we need this type of structure? You know what I mean? Like what kind of comes
that's
first?
such a good question, because it's so hard to design. Like even with a protein, with, you know, the ML tools that exist now, there are better tools to guide the design of a protein that has a desired function, but it's still a challenge.
Okay.
And with copolymers, we're like way behind. And so we had some hypotheses, like, you know, putting the acrylic acid in that structured domain, kind of like a beta sheet in a protein, is going to have a higher affinity. That panned out. But making something that's going to be selective, we can kind of guess, but we're not very good at it. And this honestly is a pretty simple system, right? We have 3 different monomers that we're putting in. They all have different roles. Like, it's a good model system, and we did this systematic study, but we still, you know, it's really hard to know what to design. And so I think that is a cool opportunity that leads to sort of what some of the rest of my lab is doing, thinking about, you know, how do we sort of leverage these statistical tools and machine learning analyses, and how do we generate large amounts of data such that we can effectively identify design principles so we know what to make.
Right. Yeah. What is like currently in the, like prior to kind of your work, like what's currently in literature? I mean, is there a database of, I don't know, like copolymer structures that, oh, we made this structure.
That's such a good question.
Like, is there like?
Yeah, there are. They are just starting.
Okay.
There's a few of them. But, you know, tools like AlphaFold rely on databases of proteins that are incredibly well maintained, right? Like, they are phenomenal. And these polymer databases, there's all sorts of other elements of complexity that you don't have with proteins.
Right.
Like, you don't have a sequence. And so they're just kind of getting off the ground.
Okay.
And so right now, it's just a very different situation from the structural biology field. But in my mind, it makes it, you know, an exciting time to be trying to learn about these types of tools and analyses.
Right. And yeah, this is super cool. So it seems like it's like, I mean, kind of like fundamental, you know, area of research, right? So it seems super exciting. Yeah, I mean, yeah, making those shapes seems incredibly difficult. Yeah, even in your model system, like what was kind of like, I mean, you had this hypothesis, but like, how'd you even start with this model system? Like, what was kind of like the thought process of making it, like starting with those monomers?
Simple.
Right.
So,
Okay.
like, I think when you're starting, you know, when you're going to any new area, you want sort of as simple as possible a model system. And so when we designed our beta sheets, like interaction directing monomer, we took the simplest possible amino acid peptide structure that's able to do this. It's been known for quite some time that 2 phenylalanines in a row is able to generate these interactions. And so we stuck it into a monomer that would be then pendant to our polymer backbone.
Okay.
We could do all sorts of more complex things. I get asked questions about this all the time. Could we strengthen our interactions? Absolutely. But, you know, there's all sorts of different directions we could go. And so with chelating to these lanthanides, you know, this is a really important question. And so that's one reason we're interested in it. But also it's very testable. You know, we put an acrylic acid in there. That's what the proteins are using. We know it's going to bind to our lanthanide ions. It's just a simple system to start interrogating these types of questions. And, you know, in some of our other work in this area, very similar type of strategy. We make copolymer catalysts, which is maybe more familiar to the synthetic community.
Yes.
But here, we are doing the most vanilla of the catalytic reactions.
Sure.
We're doing a Suzuki-Miura coupling. We have
that I'm familiar with.
yeah, like everything is as simple as possible, because what our question was how do the facets of the copolymer, so something that looks kind of like the matrix of a protein, not just the ligand, affect the catalysis. So we wanted a really simple reaction. Our ligands are triphenylphosphines. The monomer, was again basically as simple as possible to make something that we could easily copolymerize with other monomers, such that then we could really focus in on that question without too many confounding factors.
Right. Yeah, that was super cool. Yeah, I want to I definitely wanted to ask you about that paper, because right when synthetic chemist here, Suzuki Miura, that's what's familiar to me, anyways. But so like you have this self assembly of this triphenylphosphine and I guess polymer, and then palladium binds the phosphorus.
I can describe the system.
Yeah. So how does this?
Okay, so just like we took our two phenylalanines and put them on a monomer that's covalently incorporated into the backbone. My student made a triphenylphosphine containing monomer that then was covalently incorporated into the backbone. So just one of the things that we put into our polymerization mixture was that triphenylphosphine monomer.
Okay. Gotcha. Okay. And that went on to show to be, dumping a little bit of palladium, I guess, and you're able to do Suzuki Miura. I got you.
Pretty much. So we do, it's titrated, and we're careful to make sure that we don't, to the best of our ability, have a palladium that's just in solution that could be confounding our results, but add palladium.
Mm-hmm. And then something that's not really clear to me, but how do you characterize the structures? What are the techniques that are used in your lab to kind of do this? That seems like it would be difficult.
Yeah, I feel like whenever I get students from an organic background who are nervous about doing polymer chemistry, one of the things I say is that, we do the same things, right? Like, you're familiar with a round bottom flask and a vial and a Schlenk line, that's all the same thing. You're just going to get back an NMR that's more annoying.
Oh God, no.
But then, we're just asking different questions.
Right.
And so the degree to which we're able to characterize is just not the same. We're not going to get a crystal structure. As much as we and our reviewers sometimes want to have a crystal structure, that's not, right? We have ensembles. Right. And so you can take an NMR. An NMR gives you the chemical composition, and as long as your peaks aren't overlaying, and even sometimes if they are, you can use systems of equations to figure out what's in your copolymer. But then you can look at the molecular weight distribution using size exclusion chromatography. So NMR and size exclusion chromatography are our go-tos for that sort of preliminary characterization of copolymer. But then when it's in solution and you're like, is it like this? Is it like this? Then we're using scattering techniques, in addition to sometimes electron microscopy. For our assemblies where we're making micelles and vesicles, you can do microscopy and see those, which is really cool.
Yeah, I was just say.
But
how does that work then? How does that even work then? I'm not familiar with this instrument. How does that even work?
So the way that transmission electron microscopy works, which is what we use the most often, you are probably familiar with how an optical microscope works. And so if you replace light with electrons, and then your lenses, instead of being optical lenses, are now magnetic, I believe, lenses.
Okay.
It is the same concept.
Okay.
So we use a uranium-based stain. So basically the density of your atoms affects your contrast, as opposed to, absorbance and things that would affect your optical microscopy. And so we use this uranium-based stain. It binds really well to our peptides, and probably some amides in our polymer backbone. And then that allows us to visualize on this carbon-based grid what our materials, like you're physically taking pictures. But, that could have some artifacts because we're drawing them onto this tiny little grid.
Okay.
So we also look at them in solution with light scattering techniques. We use dynamic light scattering. We use my lab now does a lot of small angle scattering techniques. So you can do this with X-rays and neutrons. We mostly do this at beamlines. So Oak Ridge National Lab or Lawrence Berkeley National Lab. And then, you're using scattering data and fitting that to different equations to learn things about the size and shape of your full distribution.
That's really cool. Now, is there also like a concentration dependence? I assume there's a concentration dependence when you're trying to do light scattering stuff, but for when you're looking at, let's say, does that differ then from the systems that you're working with? So do you have to kind of, I guess a broader question would be, how does the concentration of your systems dictate the shape they form? Like, I mean, do you have to get a certain concentration? And then is there kind of like have you noticed like a breakover point before you start breaking down the structures, like in solution anyways? How does that, what's your kind of general sense?
Yeah, so fundamentally, if you have multiple chains coming together, so like with our amphiphiles, there is a critical aggregation concentration. So you need enough of them to come together and like be able to coat the interface, et cetera, such that they start actually assembling. And concentration is absolutely a factor in the sort of shape of what you make, but also whether or not it's aggregating. At some point, you're going to get to what's called an overlap concentration in solution, where like physically your polymers have to overlap.
Okay.
And so then you're dealing with a solution that is not dilute. So all of these things definitely come into play.
Right. I can imagine then, again, this is still, you're still like kind of doing fundamental stuff, but I can imagine like, is there like a desirable concentration that you mean? I assume like hypothetically, you probably want as dilute as you can to not use as much material, but I don't really know. I guess it kind of depends, I guess.
Depends on the application. Yeah. So for our catalysts, right, then you want to use as little as possible. Palladium is expensive. You have to get rid of it for all sorts of end products.
Right.
So there, we're trying to go as low as possible. For something like an industrial separation, you want as high affinity or selectivity as possible. But, in our end application, are we going to have polymers floating in solution? Probably not. They're probably going to be immobilized in some form. So, we're very fundamental. And there's like many steps towards that end application, but it is something that we try to keep in mind.
Yeah. I mean, I tell people a lot, like within academia, like we're asking the fundamental questions here. Like we're not necessarily like, pie. I mean, there are obviously going to be pie in the sky type goals, which, are great and marketable, but at the end of the day, like we have to understand the fundamental chemistry and biochemistry, what's going on here. And so it may not be as, may not be as like cool, I guess you could say, as maybe the industrial scale, but this is, I actually think this is cooler. Like we're talking about the fundamental stuff.
And because it can impact all industries. That's what I think is so cool. It's like these fundamental design principles can be important for our industrial separations. They can be important for macromolecular therapeutics. There's all sorts of different fields that you could be impacting with your research.
Right. So as we kind of wrap up here, what are some things that students or prospective students like joining your lab can expect, and also like where's kind of like the immediate future of like these projects? Like where your interest lying right now?
I think my lab now is kind of at a cool stage where I have been here for almost 6 years now. And we've established some of these platforms, right? When my first students joined my lab, I like drew some squiggles on a piece of paper and was like, best of luck. And now, I have students with this insane breadth of expertise who are helping generate these ideas. And so, they love asking this interview question, like, where are you going to be in 5 years? And Honestly, I don't know. Like, I think we're at a cool place where we've established the platforms and there's a lot of different directions that we could go.
Right.
But I think now I have the opportunity to let my students sort of help dictate where we end up. So I think, we're excited about our platforms. I am definitely excited about some of the opportunities that high throughput techniques and assay development and the analytical tools that are associated provide. So I think that's something we're going to be doing a little bit more of. But broadly, I don't know. We'll see what happens.
Yeah, we'll see what happens. Where, I guess the libraries of these, are these public? Can people just go find these libraries of kind of structures, or I don't know, or is it still kind of being developed right now?
So the databases are being developed, I would say. So the half of our lab that I didn't touch on as much is, we're trying to develop high throughput assays. So the DNA polymers you mentioned in the beginning, but I'm amused because it's our only unpublished area right now, but stay tuned.
No, it's true. I get it. It's all good.
We're trying to use DNA to generate libraries of copolymers. And so one of the questions we're really focused on is how do we rapidly generate design principles? How do we rapidly generate libraries and leverage existing characterization tools to develop new characterization tools?
Yeah. Well, Professor Knight, I want to thank you so much for coming on to the podcast today. It was extremely delightful talking with you, and I'm excited to see where this goes. I mean, I can only imagine working in this lab and making these structures and the structural relationships, and it seems like a super cool area of research right now, so look forward to it.
Well, thank you so much for your time as well, and stay tuned.
Yeah, right. All right, everyone, thank you for another episode, and we'll see you in the next one.